A rapid response team of experts from state and federal agencies and non-government organizations formed over the last week to investigate the deaths of several ohia trees in the Moloa'a Forest Reserve on Kauai's northeastern side.
And then we can decide from there who wants to go after which outliers, or if there's other trees that you see that you say, I want to sample that.
Kauai DLNR Division of Forestry and Wildlife botanist Adam Williams discovered at least 5 trees in the remote rugged forest reserve that appeared to be dead from symptoms associated with what has been called rapid ohia death, or ROD. This find set into motion the rapid response over the past week, which has included drone and helicopter survey mapping flights.
A few days ago, a few of our samples that were taken here in this area from ohia trees that showed symptoms of rapid ohia death actually were confirmed in the laboratory for showing DNA from the strain B of rapid ohia death. So we decided to come out here and get a closer look at if we find more of those trees.
Laboratory confirmation of the presence of the fungus known as Ceratocystis huliohea, formerly called Ceratocystis species B, and training for Kauai-based teams on how to identify the fungus and sample for it.
Generally, if the limb dies, it's going to hold its leaves for a while.
Lead researcher from the USDA Agriculture Research Service, Dr. Lisa Keith.
What we have recently found on Kauai is the second species called Ceratocystis huliohea, okay? We called one Ceratocystis species A, one Ceratocystis species B, with a very large collaboration with scientists here and around the world. We now have named and described 2 new species of fungi, okay? Ceratocystis species A has become Leucoohea. B is Huliohea. We now know they cause Ceratocystis wilt, or ohia wilt, and ohia canker. And it is a very different pathogen, okay? Even the genetics of these fungi are different. The first one that I described, Leucoohea, is considered a very harsh tree killer. Okay, what was formerly species B, Huliohea, we actually suspect we found because we started looking at this widespread mortality of ohia on the Big Island. Okay, so this is known as a canker pathogen, and in both cases you don't see anything on the outside of the tree.
Dr. Keith explains that the rapid ohia death moniker is a bit of a misnomer in that it does describe the Ceratocystis leucoohea species A pathogen, but includes the Ceratocystis huliohea species B, which is also widespread on Hawaii Island and now discovered on Kauai, though its impacts are much less severe and slower moving.
They go pretty much through the, into the sapwood for sure, not too much in the heartwood, but
on Wednesday, a multidisciplinary team from DLNR DOFA, Dr. Keith's USDA lab, the University of Hawaii's College of Tropical Agriculture and Human Resources, the Kauai Invasive Species Committee, and the Nature Conservancy hiked into the forest reserve to get a closer look at the 2 trees already confirmed to be infected with Ceratocystis huliohea. At the first suspect tree, Dr. Mark Hughes of USDA ARS used a hatchet and saw to peel away bark from a large multi-trunked ohia tree that has numerous dead limbs and leaves. He demonstrated to the other 16 team members how to spot telltale signs of rapid ohia death, and then how to take wood samples to send to the USDA ARS lab in Hilo for further verification.
Yes, and it's going to ask how many bags you have.
Dr. Wade Heller of USDA ARS then demonstrated how to enter data into a smartphone-based field application that tracks location, symptoms, sample collection, and photographs in a central database to ensure accurate tracking of each infected tree. After these demonstrations, field teams from KISC, TNC, and DOFA practiced sampling on the 2 trees positively identified as having Ceratocystis huliohea and 4 other nearby trees that showed symptoms. Overnight testing at the USDA ARS lab resulted in confirmation of an additional 3 infected trees for a total of 5. Tiffany Cayanini of the Kauai Invasive Species Committee said, unlike on the Big Island, not many Kauai homeowners have ohia trees in their yards, so there should be much less need for us to do test and sampling in residential areas. We do expect to be involved in continued work to investigate and test for the potential spread of the disease in the forest of Kauai. Added Melissa S.P. Fisher, director of the Nature Conservancy's Kauai Forest Program, it's fortunate that we are only dealing with the less aggressive species of this disease. We hope everyone on Kauai will be vigilant in practicing the sanitation protocols developed on Hawaii Island to keep both species from getting a stronger foothold on the Garden Island. We're looking forward to working with all the partners on next steps. Rob Hoff, State Protection Forester for Dealer-Dauphin, explained, those protocols include scrubbing boots and other footwear with 70% isopropyl alcohol after any forest activity, and thoroughly washing any equipment and vehicles that enter or are used in the forest. This should become a best practice statewide for all forest users, since we still don't fully understand how these fungi move from place to place. What is clear is that both species, and for that matter many tree diseases, enter trees through wounds, so it's important not to wound them. Since the identification of rod on Hawaii Island more than 4 years ago, it's affected more than 135,000 acres of ohia forest there, killing millions of ohia trees. The extent of the infection on Kauai is unknown, which is why more sampling will be done in the future, said Sherry S. Mann, Kauai Dauphin branch manager. And the researchers tell us Ceratocystis huliohia could have been here for a very long time, so there's no evidence that it recently spread directly from the Big Island. The initial discovery of rod has certainly made all of our people who work in the forest more aware of what to look for, and without that increased awareness, we may not have found it on Kauai and failed to take the steps we're now taking to identify the extent of the infestation and how to deal with it.